Good day. My name is Egil Marsson. I'm the CEO of Akthelia Pharmaceuticals. I'm very excited to be here today. It's my first time at LSX. Have all of you been here before? Oh, really excited to be here. I'm here to present a novel approach to battle inflammation and infections. So Akthelia was founded by 4 professors from the University of Iceland and Karolinska Institute in Sweden. The founders have been very active in the field of innate immunity and antimicrobial peptides for over 30 years. And we're one of the teams that discovered the LL37 cathelicidin antimicrobial peptide in humans, and they later demonstrated that professional pathogens downregulate the production of antimicrobial peptides in order to invade our bodies. Thus, Akthelia's motive was born to counteract the suppression of innate immunity by professional pathogens. As we all know, antimicrobial resistance in bacteria is a threat to our health systems worldwide. Infection and death rates in Europe rise by 20% annually. And the situation in the rest of the world is even more dire. Despite this, there have been no new novel mechanism of action drugs introduced to the market in the last 25 years. So there's a huge need for completely new and novel ways of battling infection. The same applies to inflammatory bowel disease and inflammation in general. Despite new drugs that have come to market in the last years, the majority of patients still do not achieve long-term remission, and surgery rates have not gone down. And this is a very interesting field, and as you see by the quotes there, it's a very attractive market as well. Maybe more attractive than infectious disease. So the problem that Akthelia is addressing is that impaired innate immunity puts it at risk for bacterial infections and inflammatory bowel disease. Pathogens downregulate the innate immunity and invade, and innate immunity and intestinal barrier are major players in the pathogenesis of inflammatory bowel disease. To demonstrate how pathogens suppress our innate immunity, on the left, we have a healthy rectal luminal epithelium expressing antimicrobial peptides. But by staining, you can see the dark area. On the right, we have the result of a Shigella infection with suppressed antimicrobial peptide expression. The epithelia is destroyed and inflammation is in progress. So this is how Shigella invades the body. The same applies to IBD in terms of innate immunity, although genetics and environment are major players in inflammatory bowel disease. The intestinal barrier and the immune response are major players in the disease. So Akthelia's solution is a family of compounds that we call APDs, arylated phenylendiamines. We are aiming at an oral therapy that restores and induces innate immunity, strengthening the epithelial barrier defenses, inducing antimicrobial peptide production, and attenuating inflammation. We are targeting the gastrointestinal tract, gastrointestinal infections, and inflammatory bowel disease. And we have a composition of matter patent for this family of compounds, which was granted last year. The compounds basically induce the production of several antimicrobial peptides, leading to elimination of bacteria. The fact that we are inducing several peptides means that the risk of bacteria acquiring resistance against a single peptide is minimized. We are inducing the tight junctions between epithelial cells, leading to a strengthened barrier. We are inducing autophagy. The compounds downregulate inflammatory cytokines, and the mechanism has been shown to be active against most microbial pathogens, including resistant microorganisms. Importantly, the treatment should leave the microbiota intact. So here is a slide that shows our compound cascade selection. We started out with a luciferase cell reporter line, which was basically screening for the expression of one antimicrobial peptide, cathelicidin. We screened around 1,200 compounds, the Prestwick library, and a number of H-stack inhibitors. With the reporter cell line, 18 compounds gave a response. We took one of them further, which is entinostat, or MS-275. After a structural activity relationship, we modified entinostat and tested 15 variants with variable concentration in the reporter line. We currently have 3 candidates for lead, which we have been testing for several cytotox, solubility, etc. And we are in the process of trying to establish a proof of concept in an established animal model, and to select one of these 3 compounds as our lead. And here we see the cell reporter line and the dose response for 10 different compounds. It's basically showing the fold increase in the expression of the gene for cathelicidin, L37. And the 3 compounds that we have selected and are studying are highlighted with an arrow. These are the compounds. The mother compound is entinostat, MS-275, and the 3 other compounds that are candidates are shown there. So the previous slide showed the result of the screening for one peptide. This slide is showing that we are actually inducing the production of several antimicrobial peptides, and also we are inducing a major ROS factor, and we are down-regulating an important inflammation marker. And in this slide, we are showing the impact on the epithelium, not the antimicrobial peptide induction, but the induction of tight junctions. So on the left, we have the result. It's an apical image of the epithelium after infection with Pseudomonas. The epithelial barrier is completely disrupted. After treatment with one of our lead candidates, the epithelium is restored. So to summarize, in an in vivo model in rabbits, you see again on the left the rectal epithelium in a healthy rabbit. In the middle, Shigella has shut down the production of antimicrobial peptides and completely destroyed the epithelium. And on the right, we have after treatment with one of the mother compound, entinostat, antimicrobial peptide production is restored, and the epithelium is also restored. So we have shown efficacy in vivo rabbit models for the mother compound, both for Shigella and cholera. And we are in the process of looking at expanding this with our lead candidates. We have shown for one of the lead candidates in vitro in epithelial cells and macrophages efficacy against Pseudomonas and multi-drug resistant Klebsiella. It's actually turning by itself. The mechanism of action, the molecular target is not known, but we do know quite a bit about the mechanism, and it's a subject of active research by our founders in their laboratories. H-stack is a very early factor, H-stack inhibition, a very early factor in the signal path, followed several hours later by a induction of STAT3, and subsequently, shortly after that, HIF-1 alpha stabilization. So we are currently pursuing a proof of concept in an established animal model and doing initial tox and PK on the lead candidates. We hope to start our preclinical talks at the end of this year or beginning of next year. We are currently funded in a pre-seed round led by angel investors from the pharma industry and with soft funding from an Icelandic state sponsor. But we will be approaching specialized life science VCs later this year and next year. So basically, Benefits and value of our approach: we are targeting directly the lack of antimicrobial peptide expression and tight junctions, leading to restoration of the epithelial barrier and resolution of inflammation. In inflammatory bowel disease, we are treating the condition that causes inflammation rather than only the inflammation itself. We are strengthening the body's own defenses, inducing a spectrum of antimicrobial peptides, including beta-defensin. If infection is initiated, the induced body's innate defenses will cure it, and resistance should not be an issue. This approach has a minor impact on the microbiota, which reduces the likelihood of relapse. We are targeting the gut with higher intestinal exposure than systemic exposure, treating the condition at site of inflammation and infection with reduced risk of adverse systemic events. Okay. So our team, myself, I do not have a background in biology or chemistry. I'm a computer scientist and physicist. I ran a medical device company for several years and exited that, spent 9 years in VC, but I'm working full-time on this company now. We have a research and development team, which is basically the founders and their labs at Karolinska and Iceland, and we have a number of advisors in terms of drug development and clinical. We are looking for advisors. We are looking for PK and PD expertise, and we are looking also for a clinical path and commercialization advice. We are thankful for our backers, and I thank you for listening.